Hello everyone, let's understand the PCR, polymerase chain reaction, the power of amplifying DNA. Why we need to study PCR? As I have discussed in molecular cloning and my previous videos in molecular biology and structural biology, that PCR is essential in molecular biology. Why? Because we need to amplify our DNA. Because DNA in very small amount is not sufficient to work with, and in biology lab you need ample amount of DNA, let's suppose either for sequencing or other experiments. So here comes the PCR. What is PCR? Polymerase chain reaction is a common laboratory technique used to make many copies, many means millions or billions copies of particular region of DNA. Okay, particular region means our DNA is 2 meter long, have different kinds of genes, but when it comes to study a specific gene, that's how we amplify that targeted genic region. We target this gene and amplify it, make it a number of copies of it. This DNA region can be anything the researcher is interested in. Okay, for example, it might be a gene whose function a scientist or student wants to understand, or a genetic marker used in forensic labs to see or to match the crime scene with DNA with suspects, or for parenting matching like paternity testing, etc. Typically the goal of PCR is to make abundant of copies of target DNA region that it can be analyzed or used in some other way. So it uses an enzyme called Taq DNA polymerase, which is a heat stable and comes from the bacterium Thermus aquaticus. PCR was introduced by Gary Mullis and colleagues in 1985, which was a breakthrough that earned them a Nobel Prize. That earned them a Nobel Prize. The technique is widely used in molecular biology to detect and analyze DNA by producing many copies of a specific DNA segments. So this is a thermocycler in my lab where I used to perform the experiments for my cloning experiments, and you can see here is the screen where you can see, I will explain you in different slide, that this is the graph. You can fix the number of cycles, etc. And this is the lid, and you put it up and place your sample inside this. So what is the working principle of PCR? So PCR works through repeated cycles of heating and cooling. This is a DNA template of your interest. It denatures at a higher temperature, which separates two DNA strands. Then there is a primer binding occurs. Primer is a short sequence of DNA which is necessary. Otherwise, how would you extend it? You want to amplify it. So from 2 or 1 strand of DNA, you get millions of copy. How? Through primer. The primer binds in the edges and it extends through Taq polymerases to synthesize new DNA. So you see here is the primer binding, then extension occur at different temperature, and Taq polymerase is preferred due to its heat stability, enabling DNA synthesis even at higher temperatures. Because for PCR, for denaturation, we need almost more than 90 degrees Celsius, and there you need primer binding, and if polymerase will not stable at that temperature, your primer will not be extended. So that polymerase, this enzyme, should be heat stable. Then PCR is highly sensitive, making it the gold standard for diagnosing bacterial and viral infection and screening for genetic diseases. Now the workflow of PCR, first what we do is a sample collection. You get the samples, what you want to work with, small DNA sample. Now you place it in a tube which is called Eppendorf tube for amplification. Further, the phases of PCR is again denaturation occurs at 95 degrees Celsius. Denaturation means separation of two DNA strands at 95 or 96 degrees Celsius. This picture has taken from Khan Academy. Okay, so DNA strands are separated by breaking hydrogen bonds. You know that DNA is nitrogenous bases of DNA is connected through each other in two strands of DNA through hydrogen bonds. So denaturation में क्या होता है that the breaking of hydrogen bond between base pairs, nitrogenous bases. Then primer binding. Then you need a primer, a short nucleotide sequence for both strands. So it is called forward and reverse primers. So क्या होता है annealing होता है इन primers का at 37 to 72 degrees Celsius. The sample is cooled down, allowing primers to bind to the DNA strands. क्योंकि DNA का एक प्रॉपर्टी होता है कि हाई टेम्परेचर पे वो ब्रेक हो जाता है एंड देन लो टेम्परेचर पे वो एनील हो जाता है. तो यहाँ पे लो टेम्परेचर यूज किया गया है एनीलिंग ऑफ दीज प्राइमर्स. नाउ elongation occurs at again this temperature. So the variation of temperature is used in this PCR and thermocycler. So 75 to 80 degrees Celsius is used to elongate this DNA, and DNA polymerase extend the primers, tag polymerase, and synthesize new DNA strands. So this is the result. After one cycle, number of DNA molecules gets doubled in every cycle. Now what happens after? Here's a stepwise breakdown of the process after PCR reaction. So PCR तो हो गया. You just put all the reagents in a Eppendorf tube, put it in the thermocycler, and after like 3 hours or so, your sample is ready to further analysis. So initially we check through agarose gel electrophoresis that either our target DNA gets amplified or not. Okay, the amplified DNA is loaded into an agarose gel which separates the DNA fragment by size when an electric current is applied. After agarose क्या होता है? We see it under UV light. After electrophoresis, the gel is placed under ultraviolet light to visualize the DNA bands, allowing the identification of size and quantity of the PCR product. Then transfer to a membrane. Blotting करते हैं उसका. The DNA fragments from the gel transferred onto a filter membrane called blotting to ensure further analysis. This can be done through Southern blotting. Analysis of DNA is Southern blotting. Then hybridization with a probe. A specific probe is used to hybridize or bind the target DNA on the membrane to confirm the specificity of the product. So these all are to specify the product and confirmation of our results. You can see how after a number of cycles you can see it in the agarose gel. Now the types of PCR. There are different kinds of PCR for different purposes. We have conventional PCR. Conventional means traditional, the simplest basic form of PCR used to just amplify DNA for analysis. Nowadays we use advanced PCR called real-time PCR or quantitative PCR, in which you can see the results in real time, okay, with the help of fluorescence. So it uses fluorescent dyes or probe to measure DNA concentration. Now reverse transcription PCR. Here what happens? It used to convert RNA into complementary DNA before amplifying it. So it commonly used for analyzing gene expression and diagnosing RNA viruses like SARS-CoV-2, etc. Multiplex PCR amplifies multiple targets in a single reaction. Usually what happens, we target a specific region of DNA. But in multiplex, you can target multiple in a single reaction using several pairs of primers. So primers are very important in PCR. You design primer through various softwares, okay, saves times and resources by detecting several genes or pathogen at once. Then coming to nested PCR, uses 2 sets of primer for increased sensitivity and specificity. Digital PCR divide the sample into many small reactions to detect rare DNA mutations. So these are the various types of PCR that used in different purposes. Now comes to very important point called CQ in PCR. So CQ is a value, or quantification cycle is a value also known as cycle threshold, CT, okay, is crucial in quantitative PCR. It represents the cycle number at which a sample reaction crosses a fluorescence threshold. That means when you have put your sample in qPCR thermocycler, then what happens after a certain point you see fluorescence. That means a light coming out that you can get through the signal in the screen. So that's what we know through CQ value. What is real-time PCR? Real-time PCR usually conducted to quantify the absolute amount of a target sequence or to compare relative amount of a target sequence. This technique monitors the amplification of the target in real time via target specific fluorescence signal emitted during amplification. So CQ is inversely proportional to the initial amount of target DNA. A lower CQ indicates a higher initial concentration, while a higher CQ suggests lower concentration of your DNA or target sample. That's how this is the graph of CQ. You can see this is a threshold. You put a baseline that okay, this is the baseline where after that a fluorescence can appear and my reaction can be visible. So low CT value gives that your DNA is of ample amount, and high CT value means your DNA is of lower amount. Okay. Some additional point regarding this PCR is there are cycle repetition after several cycles, limitations. So what happens? Amplification slows down due to reagent depletion. Reagent का कमी हो जाता है because of pyrophosphate accumulation and inhibitors of PCR like proteinase K and EDTA. And inhibitor, how do we remove the inhibitor? PCR inhibitors like ionic detergents or hemoglobin can affect results. Techniques like dialysis or ethanol precipitation can clean the DNA samples. Now coming to applications of PCR. So it is highly significant in clinical area. It has been frequently used to recognize various viral infection like infectious diseases of microorganism. Some of the viral pathogens can be detected via PCR, including human papilloma virus, SARS-CoV-2, hepatitis E, including these. And then it also used to see the quality of your samples, quality control process in the lab and lab safety. So it is very essential tool in molecular biology laboratory. I hope you have understood this video well. If you have any questions, please let me know. I will make a separate video for that. Thank you for watching.